Hello, everybody. I'd like to thank you for taking time out of your day to join us to hear more about our newest ARCH grants. We actually want to focus on two of those ARCH grants. But first, let me just make a brief note about what ARCH is. ARCH is the amfAR Research Consortium on HIV eradication. And we fund scientists, bring them together to work together towards a cure for HIV. Now, why is there so much enthusiasm lately about a cure for HIV? And the simple answer to that question is the Berlin patient. Many of you probably have already heard of the Berlin patient. We know that his name is Timothy Brown. He was an American living in Berlin with HIV, and in about 2006 he developed leukemia. And his doctors decided that he needed to get a stem cell transplantation to treat his leukemia. His doctor in Berlin made a very clever decision to look for a very special kind of donor. And that donor was a person who had a mutation that we call CCR5 delta 32. People with this mutation are very rare. They really only live in the very north of Europe. And the doctor needed to find a tissue match person with this mutation as well, and transplanted the stem cells from the blood of this donor into the Berlin patient to see if his leukemia could be cured. Now, it's about 4 or 5 years since that has happened, and lo and behold, the Berlin patient is not only cured of his leukemia, he's also cured of HIV. And that has given really a huge burst of enthusiasm and optimism to the scientific community, and a lot of us are very excited about the prospects of finding a cure. Now, the problem with the Berlin patient is we actually don't know exactly how he was cured. And that brings us to the first grant that I want to tell you about. And that's a grant that we have recently awarded to Dr. Timothy Henrich, who works at Harvard University. And Dr. Henrich has two patients who in some ways are actually quite similar to the Berlin patient. They're patients who have HIV and who had cancers. They had different types of cancers to the Berlin patient, but nevertheless they had cancers that needed to be treated with a stem cell transplant. And Dr. Henrich really did want to repeat what had been done in the Berlin patient, but if you remember, the CCR5 delta 32 mutation is very rare, and so he was not able to find a donor who had that mutation to give to his two patients. And so there are actually three very important differences between the Berlin patient and the two patients that Dr. Henrich has. One is that unlike the Berlin patient, Dr. Henrich's patients had reduced intensity chemotherapy, number one. Number two, they did not receive full body irradiation. The Berlin patient did. And number three, as I already mentioned, these two patients received a self-transplant from people who were genetically normal. They had the normal copy of CCR5-32. So they did not have the mutation that we thought was important in the Berlin patient. Now, Dr. Henrich presented these cases recently in July at the Washington International AIDS Conference, and the scientific community was very interested. I have to say that. Everybody really perked up their ears when they heard the report of these two patients, and some people in the audience even got up and said, we believe that these two patients are cured. Now, the truth of the matter is, we don't yet know if they're cured. But when I heard this talk, I too knew that I had to talk to Dr. Henrich, and I invited him to apply for an ARCH grant because we need to find out what happened in these two patients and are they really cured. So Dr. Henrich got his grant, and what he's going to do is really some very detailed studies to work out whether or not these patients are cured. He's going to do what we call apheresis. This is really an enormous blood draw, so taking out many more times than what you would normally have in a blood donation, for example. And he'll be able to extract some immune cells out of that big blood draw, and then they can infuse the rest of the blood back into the patients. And he'll be able to take a close look at those cells from the blood to see whether or not they contain HIV. He's also going to take tissue biopsies from what we call the gut. In this case, it's actually going to be from the rectum. Now, you may be asking yourself, why would we go there? It actually turns out that regardless of the route of HIV transmission, the great majority of HIV resides in and around the rectum. And so if you're going to find any HIV in these people, you're probably going to be able to find it there. And finally, he's also going to look at the cerebrospinal fluid, because this will give him an indication of whether or not there's still any HIV in the brain, for example. It's a sanctuary where antiretroviral therapy doesn't reach as well, so it's possible that HIV could hide out there. So this is going to be one way for him to look at whether or not there's still some HIV there. He's also going to take the patients off their antiretroviral therapy. This really is the ultimate test to determine whether or not a person is cured of HIV, because under normal circumstances, if there's any HIV left, it's going to come rebounding back in the absence of antiretroviral therapy. So when he takes these patients off antiretroviral therapy, which of course he's going to do under very controlled conditions, he's going to be closely monitoring these patients to see if and when any virus comes back, but this is really going to be the ultimate test of whether or not these patients are cured. Now, we're going to know the answers to these questions probably by next September. This is a one-year grant that Dr. Henrich is doing, and it's going to be super exciting to find out, are these the next two Berlin patients, or in this case, of course, they would probably be the Boston patients. And it's going to teach us a lot about what does it take to cure somebody. What do we need to look for? What kind of procedures does a person need to go through in order to find a cure for HIV? Now, the other grant I want to tell you about, the other new ARCH grant, we have 4 altogether, but I just wanted to concentrate on 2 today. The other grant goes to Dr. Deborah Prasad, who works at Johns Hopkins University. And this grant actually came about from a small meeting that amfAR convened at the University of California, San Francisco in June of this year. And we convened that meeting because we wanted to know specifically what is it going to take to cure HIV in kids. These are people who acquired HIV perinatally, which is the word we use when we say that the HIV is transmitted from the mother to the child around the time of birth. So Dr. Prasad reported to us at that time that she and her colleague, Dr. Catherine Luzuriaga, who works at University of Massachusetts Medical School, between them have 5 patients who were infected perinatally. Now, standard practice is that we give antiretroviral therapy to newborns who are born to HIV-positive mothers. And that is done in an attempt to prevent HIV transmission. And these days, that treatment is very successful. There's a very high rate of success in preventing HIV transmission. But this was actually done in the mid to late 90s, at the time when we hadn't really quite perfected the antiretroviral therapy under these circumstances. And so unfortunately in these 5 kids, HIV infection did take hold. But what it does mean is that these 5 kids have been taking antiretroviral therapy since soon after birth. And now a newborn has a very different immune system from an adult. And it does appear that some very remarkable things have happened in these 5 kids. We know, for example, that they no longer test HIV positive, which is not necessarily terribly unusual in kids like this. But even in the detailed tests that Dr. Prasad and Dr. Luzuriaga have conducted so far, they haven't been able to find any HIV. And obviously, we and others are asking ourselves, is it possible that these 5 kids are cured? So along similar lines to Dr. Henrich, we decided to award a grant to Dr. Prasad to find out whether or not these kids are cured. They have actually mustered together a team of probably 10 scientists or more who are each going to apply their very specialized techniques, looking for any way that they can find a trace of HIV in these kids. It is actually a little bit more challenging than when you're doing these kinds of tests in adults because kids, by their nature, are smaller. You can't take as big a blood draw. It's much more difficult to do tissue biopsies, and so they're actually adapting a lot of the tests that we would use in adults and applying them to kids to find out whether or not there's any HIV in there. Now, there are lots of reasons to be excited about this particular grant. One reason that amfAR is particularly excited is because I think it ties in nicely with some early research that amfAR funded in the early 1990s that actually laid the groundwork for using antiretroviral therapy to prevent HIV transmission from mother to child. It was studies that we funded in the mid to early 90s that laid the groundwork for scientists to understand how it is that you could give the antiretroviral therapy both to the mother and to the newborn to help prevent the transmission. And so we think it ties in very nicely. We're very excited about that. And another reason we're very excited is because we're used to thinking that antiretroviral therapy cannot cure HIV. Now that's probably true for the vast majority of people alive today who are HIV positive. But what we're seeing a glimpse of here today is the possibility that in these kids under these very specialized circumstances, maybe it is possible to cure HIV with antiretroviral therapy. You know, I think these 2 grants really highlight some very novel ways in which amfAR is delving into the landscape of cure research and really pushing our boundaries of what we understand and how we think about what it's going to take to cure HIV. Now, I'm just about to wrap up on the description of the grants, but I did want to make a couple of final notes about ARCH. We think ARCH is a great program for finding a cure for HIV, and we've implemented it because we think it has some advantages over the ways in which research is traditionally funded. Certainly at amfAR, we have a very much faster turnaround on grants than they do with the NIH, as an example. So the two grants I told you about today, Dr. Prasad presented her findings in June, and Dr. Henrich presented his findings in July, and within 2 or 3 months, we were able to actually get funding out the door to those two researchers to pursue what it is that they've found, to pursue what it is that seems so exciting, that seems to put us on the cusp of finding a cure for HIV. Another thing about ARCH is we're able to direct the scientists to the questions that we know are the exciting ones, the ones that have the promise in bringing us forward. And a third way that we pride ourselves on how ARCH works is that we are able to organize scientists in a way that, frankly, they're not very good at themselves, nor do they really much enjoy. Now, I know we have some scientists on the phone, I apologize, but I think we recognize that the strength of scientists is that they really want to just be doing the work that they do. And to be honest, at amfAR, that's exactly what we want the scientists to be doing. We are then in a position to be able to organize how the scientists come together and how they organize their projects to dovetail in with one another so that the sum is really greater than the parts. What we do is we bring together these great minds and make excellent strides. We've already made huge progress in bringing to clinical trial a drug that nobody ever would have thought might play a role in a cure. We've brought together scientists who are comparing and contrasting ways that can measure the teeny tiny amounts of virus that are pertinent when you're looking to find a cure for HIV. And in an example I just told you, we brought together scientists looking to see whether it's going to be something very different to cure kids. To sum it all up, we are very excited about ARCH. We hope that you are too. And I do believe that we might be taking some questions.
Yes. Thank you very much, Rowena, and we hope you are all as excited as we are about these projects. If you would like to ask a question, if you have called in over the phone, you can now press *1 on your phone. If you hear 2 beeps, that means that your line has been unmuted and you can go ahead and ask a question. If you are viewing this over the webinar, you can click the little hand icon to raise your hand, and then you can type your question into the space provided below. I'll be reading those questions out loud, and I think we're going to take our first call from the conference call. And if you just heard 2 beeps, go ahead and ask your question, please.
Yes, hi. I have a question about the Boston patient as opposed to the Berlin patient. If the stem cells that the Boston patients received did not have the same special mutation that the Berlin patient's stem cells did, why exactly would this method work to cure their HIV?
That's a great question. I'm just going to repeat it to make sure everybody heard it. There's a key question as to the Berlin patient got cells from a donor that has a CCR5 delta 32 mutation, and yet the Boston patients got cells from people who have just a regular copy of that gene. So how is it possible that the Boston patients might be cured? And frankly, this grant is actually aimed at finding out why. I can tell you that Dr. Henrich thinks it may have something to do with 2 particular factors. One is that his Boston patients, during the whole process, and indeed until this day, are taking antiretroviral therapy. That was not true of the Berlin patient. So these Boston patients, they received their transplant. During that time, they kept taking their antiretroviral therapy. Even 2 to 4 years after their transplant, they're still taking that antiretroviral therapy. And we think that when those cells were transplanted into the new patients, they would have been susceptible to new HIV infection, except that these patients were taking antiretroviral therapy, and that prevented the ability of the virus to spread to those new cells. The other factor that's probably important here is what we call graft versus host disease. And this is kind of a little bit of an unusual situation. People are used to thinking of, when you receive a transplant, you're used to hearing about people might reject that transplant. If you get a liver transplant, a heart transplant, we're familiar with the concept that you might reject the transplant. But it turns out that with these particular types of transplants, when you're transplanting stem cells from the blood, because they are essentially an immune system into themselves, when they get transplanted into the host, they can actually reject the host. they can actually do a little bit of attacking of the host. Now, we're used to thinking of it the other way around, that the host would attack the transplant. This is the transplant attacking the host. And when it does that, it might specifically, or at least inclusively, kill the cells that were in the host that were infected with HIV. So the transplant might actually be doing double whammy duty in not only replacing the infected cells, but killing any of them that might be remaining. But, frankly, we're going to find out better answers to that in about a year's time when Dr. Henrich has finished his research.
Great. We are going to take a webinar question now. This question is, you mentioned that HIV hides in the rectum. Is this a viral reservoir?
It is a viral reservoir. We think of the rectum as a tissue sanctuary, if you like. And it's, like I mentioned before, it's regardless of what route of transmission. If you were an injection drug user and acquired HIV that way, if you acquired it sexually through the vagina, if you acquired it sexually through the rectum, in all of those cases, HIV tends to concentrate in the rectum. And the reason for that is it so happens that the very cells that HIV likes to live in and feed on are most highly concentrated in the rectum. And so that's where they're getting all of their fuel and all of the places that they like to hide out and live. And so the virus does tend to congregate there and specifically concentrate in that area of the body. So that if you're looking to see whether or not somebody's really cured, the best chances you have of finding the HIV is if you look in the tissue of the rectum, because that's where it's most likely to be if there's just a little bit left.
Great. We have time for one more question. This question, also from the webinar, is, of the children who have appeared to be cured, has their immune system in effect adapted to their HIV?
Also a very interesting question. Has the immune system of the children adapted to HIV? Now, one of the things that you've probably heard of, when children are born, they actually have a very, very immature immune system. They're not fully developed at all, and that's why that under most circumstances you would recommend that a woman breastfeed, because a lot of the very antibody-rich milk comes out of the woman's breast certainly the first few days after birth, and it gives a boost to the immune system of the infant. Now, what that means is that if the infant acquired HIV during the birth process, then the HIV is actually entering into a body that essentially has almost no functioning immune system, or to put it better, it functions a very different way than the way an adult immune system does. And so sometimes, especially if the child goes immediately onto antiretroviral therapy, which then brings the levels of the virus very, very low, the body doesn't really detect that the virus is in there, and so under some circumstances, the infant never develops antibodies because the body doesn't detect that there's anything that needs to have antibodies made against it. And so this is just one example of how very different the immune system is between an infant and an adult, and it's why we think it might take something very different to cure an infant and an adult. And nobody else that we had ever heard of had really looked in detail into the question of, well, of course it's difficult to cure an adult, but we're also going to want to cure kids. And amfAR took the lead on this question, and we're going to find a lot more answers to what happens in infants who are infected perinatally through this collaboration that Drs. Prasad and Lazuriaga have set up.
Great. Well, I think that's unfortunately all the time we have. Thank you, Rowena, so much for doing this. And thank you, everybody who called and logged in. If you had a question that we unfortunately did not get a chance to get to, please feel free to email the person that you RSVP'd for this call to, and we will do our best to get that answered for you. Thank you again.
Thank you, everyone.